Okay, so this is DNA translation. So we have just our DNA strand right here. This top strand is the template strand, and the bottom strand is the coding strand. We're only going to be looking at the coding strand today because these are complementary bases, and this is going to be perfect to use for our translation. So we have a bit of a problem here. We can see we have thymine in this RNA strand, which is incorrect because there is no thymine in RNA. It's uracil instead. So we need to trade all of these T's for U's. Now we're ready to go to start with translation. So we can see AUG, our start codon right here, or methionine. And now we're just going to code everything in triplets to get our amino acids. And we get valine, arginine, phenylalanine, and lysine, and then our stop codon. So our first type of point mutation is a missense mutation. And this is basically whenever you're exchanging and putting in the new nucleotide, you're going to go from one type of amino acid to a new kind. So we add in this cytosine right here, and that brought us from arginine to now we have serine. Next, we have a silent point mutation. And this is where, even though we're adding in this adenine nucleotide right here, we're still going to get the same lysine amino acid that we got before. So you're just basically staying within the same amino acid. You're not changing. Next is a read-through mutation. And this is whenever originally we had a stop codon right here, but now since we added in a new and different type of nucleotide, we have just a normal amino acid and translation can keep going. So originally this was a stop codon and now it's tyrosine. Finally, we have the nonsense mutations. And this is where we have just a normal amino acid, and then that gets turned into a stop codon after we put in the new nucleotide. So this used to be tyrosine, but now it's a stop codon. So just a brief review, this is a missense point mutation where we go from arginine to serine. We are changing amino acids. And this is a transversion because we went from purine to pyrimidine. Next is a nonsense. And this is where we have an amino acid that gets changed into a stop codon. This example is a transition because we went from pyrimidine to pyrimidine. Next is a silent point mutation. And this is where we stay with the exact same kind of amino acid. We had lysine, and then we had lysine again after. And this is a transition because we had purine to purine, same type of nucleotide. And then finally is a read-through. And this is where we go from a stop codon to a normal amino acid of tyrosine. This is a transversion because we went from a purine to a pyrimidine.